Hi, today we are going to be talking about designing CRISPR guide RNAs to correct a single point mutation responsible for sickle cell anemia. In this project, we will utilize sequencing and design tools such as GenBank, Genius Prime, and ADAP to identify a sequence for a healthy HBB gene, as well as a mutated HBB gene, the one responsible for sickle cell anemia, and use these sequences to design a gRNA and repair template that will effectively allow for homology-directed repair to correct a single mutation. CRISPR-Cas9 is a gene editing technology that is anticipated to assist in many scientific advancements. Gene editing has immense potential with genetic disorders, as it can fix problems at their roots, unpreferable gene sequences, rather than just treating their symptoms, which would be the continuous production of maladaptive gene products. CRISPR utilizes an artificially designed guiding RNA, gRNA, to target the designated sequence complementary to the gRNA. There, it makes a cut in the sequence, which causes a double-stranded break. Deletion may occur through the non-homologous ends joining. Here, the DNA joins together the two disjoint strands of DNA. CRISPR may also be used to insert or correct sequences when using a repair template. A repair template is used in homology-directed repair. Here, homology arms that correlate to the surrounding DNA sequence, along with a target change in the sequence, are used as templates for repair. This repair structure is introduced to the cell in a mass amount in order to encourage the cell to use the repair template for repair of the cut sequence site rather than the sister chromatid.
So the CRISPR gene editing technology is widely popular for its potential to cure diseases. One of the most widely discussed examples in this regard is sickle cell anemia, a devastating blood disorder which is a severe hereditary form of anemia, in which a mutated form of hemoglobin distorts the red blood cells into a crescent shape at low oxygen levels. Bone marrow transplant was the only real treatment for afflicted patients before CRISPR gene therapy. Sickle cell disease is one of the most common diseases and affected over 30,000 infants a year. Sickle cell disease is used to describe a group of hemoglobinopathies that have a mutation in the gene that encodes for the beta subunit of hemoglobin. It is made of 4 globin subunits, 2 from the alpha globin and 2 from the beta globin. Sickle cell anemia arises when there's a single base mutation that arises in the beta globin gene. This change is typically glutamine to valine in the 7th position and causes a structural change in the hemoglobin.
So the way that we went about this is that we first had to find the sequence for the beta globin gene as well as the sickle cell variations. Many of the variations we found were all single base mutations that caused the GLU7 mutation, which is glutamine to valine at the 7th codon. After we identified these sequences, we took these sequences and transferred them to Genius Prime, a DNA editing and analysis tool, and we're able to compare these two sequences and confirm there was only a single mutation. These sequences were then taken and put into ADAM, a guide RNA design tool, and here many viable variations were found as listed below in our table. Higher scores mean that there are a less likelihood of off-target effects, so we chose the first one as it has a score of 100%. And then finally, we made a repair template with those 40 base pair length homology arms on each side of the cut sites and the target site. And the GAG sequence for the 7th codon was present in order to correct that GTG sequence. As you can see in our results, in our first figure, we have that change in the sequence at the 7th amino acid. And in our second figure, we show you where our guide RNAs target and cut. And then our third figure shows you what our repair template will be like in order to correct that sequence. Overall, the aim of this investigation was to identify and annotate a guide RNA useful for targeting the GLU7 val mutation that occurs in the HBB gene of those with sickle cell anemia. We also designed a repair template in order to encourage homology-directed repair off the repair template in order to correct that change, that mutation. Overall, this could be a really successful tool to use. However, there have been some concerns using CRISPR. Recently, sickle cell anemia was actually treated in a clinical trial, but that clinical trial was halted due to 2 of the 14 patients developing blood-related leukemia. This draws some concerns as to what the mutation rate of CRISPR is when in the body for long periods of time, and more research needs to be done about the off-target effects that CRISPR has in order to ensure the efficacy and safety of using CRISPR on humans for treating mutational diseases. Thank you.